Here I want to make another short parenthesis, because as I mentioned at the beginning, by chance I started to work in diagnostics because I had some experience that I got when I worked with coffee at the lab of Heart, and I started to receive many samples from different parts of the world. And I analyzed different types of samples. So soil samples, sometimes we got the fungus itself, parts of the plants, and we started to act as a diagnostic center for tropical race 4, and that led to the publication of several first reports in which I was responsible, let's say, for this work, for this part of the work. So Jordan, Lebanon, Vietnam, Laos, Myanmar, and unfortunately Colombia. So as you can see here, also different techniques were used some years ago. There is an evolution also in terms of time, because when we started here with the samples of Jordan, we have to do these FCGs, which takes a lot of time, but now we just sequence. So of course there are some more resources here, and this is what we do now. There's been also in the last years an evolution in the techniques to detect TR4. So initially we just by eye check first in the field, compare with the symptoms, and then the FCGs, the PCR, real-time PCR, and the last thing that we have, at least here in the Netherlands, is the lamp, which essentially can be used in the field directly next to the plants, which in theory is not as easy as that, but is quite close to reality. So I use it in Colombia. Hert and his colleagues developed this what they called the forensic analysis, which allows you to at least give an interpretation of the possible origin of the incursion by looking at the relationship between strains. So for example, in this case, even though you can see here that all tropical race 4 are from a same clades, like a clonal lineage, they have some difference based on the SNPs. And in this case, you can see, for example, that the strain from Pakistan is closely related with Philippines, Jordan with Lebanon, and of course the one from China to Myanmar, Laos, and Vietnam. I also work indirectly with diagnostics in other fields. And for example, I help James Dell with the transgenic Cavendish bananas to see if those plants are really resistant. So we use our diagnostic techniques and the protocols for inoculations with these plants. We also help the group of Martin Rep in Amsterdam with his studies about the 6 genes. And of course, also many companies that want to try products like fungicides, for example. So that, thanks to that, I've been invited several times to Latin America to different labs to teach in different workshops related to diagnostics of tropical race 4. We wrote this book last year. It's in Spanish, but of course the English version is coming soon. You can download it from my ResearchGate.